Hello, today we will be continuing with our unit on Evolution Explains Life's Unity and Diversity with the guiding questions: what evidence supports Darwin's theories of biological evolution? And how do evolution and natural selection explain life's unity and diversity? So, to tie this in, we're going to start with a question from what we learned previously about how dogs were selectively bred from wolves. So, through the process of artificial selection, which is similar to natural selection, except instead of the environment deciding which traits are selected for, humans decide which traits are selected for. So, as you're answering these guiding questions throughout the notes and the lectures, I want you to try to approach them from the four aspects of the evidence for evolution that supports natural selection and evolution, which are the fossil record, comparative anatomy, molecular biochemical evidence, and comparative embryology. So, as you're answering these questions, you want to look at all four of them and see if you can make them apply to the question. So, is there evidence in the fossil record for dogs being derived from wolves? Are there similarities in their body parts? Is there DNA evidence to support that dogs were derived from wolves? And is there any similarities in the embryological development while the pups are being formed in the womb? So, one of the questions that was posed this week was specifically talking about English bulldogs. English bulldogs have been so selectively bred that they have an alarmingly low genetic diversity within this breed. So many of them have a lot of health problems that may or may not be able to be reversed, since the genetic diversity within this breed of dog is so limited due to human interference and selectively breeding throughout the generations. So as you decrease genetic variation within a population, you tend to see health problems starting to arise. So, who are some of the pioneers in the field of genetics? Well, first we'll talk about Gregor Mendel. He was a monk that did experiments with heredity by studying pea plants. So he crossbred yellow and green pea plants and smooth and wrinkled pea plants with known traits, and he studied what happened with the offspring generation after generation. So through that, he was able to come up with the principle of independent assortment, showing how genes travel from parent to offspring. Reginald Punnett helped us develop a tool that we can use to predict the probability of offsprings that we will be using during today's lecture. So again, heredity studies how traits are passed down from parent to offspring. Diploid organisms that have two parents have two copies of each gene. During a process called meiosis, those two copies are separated, where you get one copy each that's passed on to the next generation or the offspring. We will talk about meiosis at a later date. So just know that you get two copies, one from each parent. So DNA is the material that carries information about living things. DNA code has been passed down from species to species and is part of the evolutionary evidence, because the same code of the base pairs and nucleotides is the same in all living things on Earth. So each piece of information that actually codes for something in the DNA, we call a gene. So genes are responsible for creating the genetic code to make a protein. Proteins are what give you your characteristics and traits. So DNA has genes, genes code for proteins, proteins give you your traits. Now you get two different copies of each gene that codes for a specific protein, one from each parent, and those things can be the same or different. The different alleles or the different copies of a specific gene is what provides for a genetic variation within a species. Now, if the two alleles that you inherited for a specific gene are the same, then we say that you will have a homozygous copies of those genes. So you have two of the same. In genetics, we use letters to represent genes. So in here we have two capital B's or two little b's, which are homozygous. The prefix homo means same. Heterozygous means you have two different copies. The prefix hetero means different. So in this example, you have one large B and one small b. So in the example that we'll be using today, remember an allele are different copies of the same genes. So our two different alleles that we'll be discussing are for feather color in birds. So lowercase b will represent the blue recessive gene, and uppercase B will represent black, the dominant gene. When we're talking about recessive or dominance, that means that one gene is expressed whether you have one or two copies. We say it to be the dominant gene. Recessive genes are expressed when you have to have two copies of it. Phenotype is the trait that is expressed. But before I get to phenotype, let me discuss genotype first. Genotype is just simply what two copies did you get. So did you get two of the same, two different ones, or what are they? That is your genotype. Your phenotype is based on your genotype, and it determines what is expressed. In our example, it will be blue versus black. So we're going to be using the Punnett square as a tool to be able to predict what our phenotypes and genotypes will be when we cross different genotypes. So let's get started. In our first example, like Mendel, we're going to start off with homozygous parents. So we're going to have a homozygous dominant blackbird crossed with a homozygous recessive bluebird. So remember, you get one copy from each parent. So when using the Punnett square, you put one parent's contributing allele on the left side. And then remember, you're also going to get one copy from the other parent, and you write those along the top. When using the Punnett square, for the parent on the left, you're going to fill in that trait going across the Punnett square like so, for both the top and the bottom. Then after you're done with that, you're going to fill in the boxes also with the alleles of the parent that's represented at the top by filling it in from top to bottom on the left and then the right. This gives you your combination, the possible combinations of the offspring. Now that we've done that, we can figure out what our genotypes and our phenotypes will be. Remember, genotype is simply the allele copies that are present. So they are represented with the red letters here that are circled. So for this one, since the parent on the left only had a capital beta offer and the parent on the top only had a lowercase beta offer, the genotype for all four of the offspring will be big B, little b, and they are heterozygous because they have two different copies. And the probability of the offspring will be 4 out of 4, meaning 100%. All of the offspring of this cross will be heterozygous, big B, little b. Now, what does that tell us about the phenotype? So this is the trait that we will actually see. So are birds going to be blue or are they going to be black? So since they all have at least one capital B in them, which is dominant, then all 4 out of 4 will be black. And also the phenotype for when you cross a homozygous dominant with a homozygous recessive, you get all 4 birds to be black. Now let's try a different cross where we take the heterozygous offspring and we mate them in the next generation. So we're going to mate 2 heterozygous ones. Now, again, remember, one parent goes on the left, and they have one copy of the allele to contribute. So we're going to put the 2 different possibilities that this parent can contribute, one on the top, one on the bottom on the left. And then the other parent has one big B and one little b also to contribute. We're now going to fill in the parent that's written on the left from left to right in each box, as well as at the bottom. Now we're going to take the alleles of the parent that's represented at the top and fill in top to bottom on the left and then in the right. Now this gives us our possible genotypes and phenotypes again, but now it's different than last time. So this time we have allele copies. We have big B, little b, little b, little b, and big B, little b, and big B, little b. Now, just like in math with the communicative properties, these two are the same. They both have a big B and they both have a little b. It doesn't matter if one's on the left, it doesn't matter, and the other's on the right. They're both big, one little. So these are both heterozygous. So to get this genotype from this cross, 2 out of 4 or 1 out of 2 for the ratios, or 50% will be heterozygous. Also have 1 out of the possible 4 that is homozygous dominant. So 25% of the offspring of this cross would be homozygous dominant. Then also you have 25%, 1 out of 4 will be homozygous recessive. These are our genotypes. So from crossing 2 heterozygotes, we have 3 possible genotypes that can be created. Now, phenotypes. What are our different phenotypes? Trait that we will actually see. Only one out of the 4, the homozygous recessive, will be blue because it has 2 copies of the BB. Although 3 out of the 4 phenotype will be black. Because remember, you only have to have 1 copy of the dominant gene for it to be expressed. So that's how we got the 3 out of the 4 would be black. Because this has a big B, this has a big B, this one also has a big B. Now let's do another example. What happens if we cross a heterozygous with a homozygous dominant? So again, remember, you can get 1 copy possible from each parent. So we write those 2 possibilities. But now we're going to switch over to represent them with letters, which is what you'll do in your Punnett squares. So instead of drawing the pictures of the genes, now we will have the actual letters. So the red parent is going to be written on the left side, and the purple parent is going to be written at the top. So just like before, you're going to fill it in each box. The parent on the left is going to go across, and the parent on the top is going to be filled in going down. After we do this, these are our possible genotypes that we get and our possible phenotypes. So now let's combine like with like. You see, when we have a heterozygous versus a homozygous, we get 2 different possible genotypes. Either for this one, dominant BB, so these birds will be black. Or we also have 2 heterozygous, 2 out of 4. So both for the homozygous dominant, the probability is 2 out of 4. And for, the homozygous dominant, the probability is 2 out of 4. And the heterozygous is 2 out of 4. Now the phenotype, this one has a capital B, so does this one, and this one also. So all 4 possibilities have a capital B, so all 4 of our phenotypes will be black. What happens when we cross a heterozygous with a homozygous recessive? Then we get our letters again. This is going to go on the left. And our homozygous recessive, we're going to put on the top. When you cross these are our possible genotypes and phenotypes. And when we combine the information together, then 2 out of 4 will be little b genotypes, and 2 out of 4 will be big B, little b genotypes. And then for the phenotype, 2 out of 4, the top 2 here will be black. And then for the bottom, I messed up. This should be blue. So let me correct that really quickly. Sorry about that. Okay. So then again, let's go through this again. So there'll be black on the top, and blue on the bottom. All right. So now let's practice. Indicate whether the genotype is homozygous or heterozygous. Remember, if the two are different, meaning one big, one little, then it is heterozygous. And if the two are the same, whether it's too big or too little, then it is homozygous. So this would be our answers from the top. So please check your notes and see if you got those correct. And then if black hair is dominant over blonde hair, then that means you just need one copy of the dominant for it to be expressed. So our answers for this would be black, black, and blonde, because these two have a copy of the dominant and this one does not. So the recessive trait is expressed. Make sure you go through the vocabulary. This is a very vocabulary rich topic, so make sure you copy these into your notes. But you can use the same Punnett squares to figure out incomplete dominance and codominance, which are two things that are similar to dominant and recessive, but not quite. So in codominance, unlike in dominant and recessive, where one rules over the other, codominance means both alleles are simultaneously expressed, like in humans with the ABO blood system. A and B are codominant. That's how we get the blood type A and B. So in the heterozygous goats of codominance, then you have both expressed. So in this example of genotypes and phenotypes for dogs, let's say pink tongue versus black tongue, if you have a heterozygote and it's spotted pink and black, then we would say that that is codominant. Also, black versus brown, if you have black and brown in the heterozygote, then that is codominance. Both are expressed. Also, with black fur versus white fur, if you have, say, a cow pattern with black and white fur, then that would be codominance. The other alternative is incomplete dominance. It's when both phenotypes are expressed as an intermediate, so they're not fully expressed. It's like a combination of the two. So instead of being short versus long, you're average, or I'm in the middle. Long versus short, you have medium legs. Same with fur length, long versus short, you have medium. And curly versus straight, you have curved. So like with hair, curly hair versus straight hair, the incomplete dominance would be wavy hair. So I hope this helps you with the vocabulary for this week, and good luck studying.